Let's look at dipole-dipole forces in more detail. As we mentioned before, dipole-dipole forces are displayed by polar molecules. In an early slide of this lecture, we showed a cartoon, which we reproduce below, illustrating the intermolecular force involved when two polar HCl molecules are near each other. Each HCl molecule is considered a dipole, having two opposite poles, a partially positive and a partially negative end. And because of the polar nature of the HCl bond and the linear shape of the molecule. And because each of these molecules is polar when they're near each other, there's a dipole-dipole force exhibited between the partially negative end of one molecule, in this case chlorine end, the partially positive end of the other molecule, in this case the hydrogen end, and we represent it by these red hash marks. It would seem reasonable that more polar molecules, meaning larger magnitudes of the partial positive and negative charges, would exhibit stronger dipole-dipole forces, and less polar molecules, meaning smaller magnitudes of delta plus and delta minus, would exhibit weaker dipole-dipole forces. Conveniently, there is an experimentally measurable quantity called dipole moment, represented by the Greek letter mu, and measured in units called debye's, or just abbreviated as D. These experimental quantities can be obtained in a lab and tabulated, so you and I can look them up. And they can be considered a measure of molecular polarity. In other words, the larger the dipole moment, the more polar the molecule. If the molecules we're talking about are just simple two-atomed molecules like HCl, composed of two atoms, and we know the bond lengths, the distance between the center of the hydrogen and the chlorine atom, we can actually calculate a number for the partially positive and negative charges on each end of the molecule. So the table below has not just the simple molecule HCl, but the cousins of HCl, which could be considered hydrogen bromide and hydrogen iodide. And these molecules become larger as you go from chlorine to bromine to iodine. So for example, the bond length goes from 127 picometers in HCl to 161 in hydrogen iodide. The electronegativity difference between the two atoms in HCl is the largest. It's smallest in hydrogen iodide. If you do the experiment and measure the dipole moments of these molecules, we find that the dipole moment of HCl is 1.08, that of HBr is 0.82, and that of HI is 0.44. This isn't surprising because the bonds in these molecules are becoming less polar as the electronegativity difference between the two atoms becomes less. The point here is that the dipole moment values tell us that HCl is the most polar, HI the least polar. If you use the bond length and dipole moment data correctly, you can actually calculate a charge for each end of each of these molecules. So in other words, in HCl, it's calculated that the partial positive charge on the hydrogen is plus 0.178. The negative charge is minus 0.178. And you notice these partial charges on either side of the molecule become less as the molecules become less polar. Dipole moments drop. And so we say that as the measured dipole moments change, the calculated partial charges on either end of the molecules change. My arrow here is going up. So we'll say it in the opposite way. As the dipole moments increase, the calculated partial charges increase. So obviously, HCl is the most polar molecule in the set. HI is the least polar molecule. If the intermolecular forces increase due to increasing dipole-dipole forces in the series HI, HBr, and HCl, we would expect to see the measured boiling points of the liquid forms of these molecules increase as these dipole-dipole forces increase. The table below shows both the dipole moments and the measured boiling points of this series of molecules. So again, we say HCl has the largest dipole moment. It's the most polar molecule. So the dipole-dipole forces between HCl molecules should be the strongest. HI, which has the smallest dipole moment, should have the weakest dipole-dipole forces. But something here is not right. You'll notice that the observed boiling point of HCl is the lowest, and the observed boiling point of HI is the highest, even though the dipole-dipole forces in HI are the lowest. So something is not right here. Well, perhaps now is a good time to recall that all molecules experience dispersion forces. Don't forget dispersion forces. And these might be playing a dominant role here. Remember, dispersion forces tend to increase with increasing molar mass and larger and more polarizable electron clouds. The table below shows the molar masses of these three molecules along with their boiling points. Now, this table makes sense. The lightest molecule, the one with the smallest molar mass, has the lowest boiling point. The one with the highest molar mass has the highest boiling point. That makes perfect sense. So, what's going on? Well, we will soon see that we were correct to expect that the most polar molecule, HCl, should have the highest boiling point. However, we would only expect that if we were only considering dipole-dipole forces. Remember, we can't forget dispersion forces. In this case, the dispersion forces operating dominate. So HCl has dipole-dipole forces and dispersion forces. HBr has weaker dipole-dipole forces but stronger dispersion forces. HI has the weakest dipole-dipole forces but the strongest dispersion forces, and in this case the dispersion forces rule, they dominate. We have to keep this in mind. So the take-home lesson here is that we need to consider all possible types of intermolecular forces when making predictions about the expected chemical and physical properties of substances. In some cases, one particular type of intermolecular force could dominate. Again, in this example, these are all polar molecules, but they all have dipole-dipole forces, but those dipole-dipole forces are not calling the shots. Here the dispersion forces are calling the shots. Now on the next slide, we will attempt to isolate the effects of dipole-dipole forces by attempting to keep dispersion forces constant. So how will we do that? Well, here's a large table of data, and let's look and see what's in this table. We have four molecules: propane, dimethyl ether, acetaldehyde, and acetonitrile. In the second column, we have ball and stick models of these. So for example, propane has 3 carbons, which are the gray spheres. The N carbon has 3 hydrogens, the middle carbon has 2, the other end carbon has 3 hydrogens, and so forth. And then we have a space-filling model of propane. Dimethyl ether has 2 CH3 groups, which are called methyl groups, with an oxygen in the middle. Here's the ball and stick model, the space-filling model. Dimethyl ether is polar. You'll notice propane, we would consider propane to be nonpolar. It has a very tiny dipole moment, very close to 0. Dimethyl ether, because of the very electronegative oxygen, does have partially positive and negative parts of the molecule, and it has a measurable dipole moment, so it is polar. Acetaldehyde has a different structure again. Here's the space-filling model, and it has a even larger dipole moment, and an acetonitrile containing the nitrogen. Quite polar molecule. Here's a space-filling model, and at least relative to some of these other numbers, a fairly high dipole moment of 3.9. So we say that the dipole-dipole forces are increasing, going from propane to dimethyl ether to acetaldehyde to acetonitrile as measured by the dipole moments. What we've done here is we've picked 4 molecules that have pretty much the same molar mass, not exactly, but pretty close. So what we're going to say is that we're taking a shot at keeping the dispersion forces relatively constant because the molar masses of these are very, very similar. However, the dipole moments are increasing quite dramatically. So we say the dipole-dipole forces should increase. So let's hold dispersion forces constant relatively and make the dipole-dipole forces increase. Now look at the boiling point data. This makes sense. We say we've kept the dispersion forces constant. Now we can see that yes, as dipole-dipole forces increase, as reflected in the increasing polarity of these molecules, the boiling points do increase quite significantly. Let's look at hydrogen bonding in some detail. Remember, hydrogen bonding is not an actual bond in the sense of a bond like a single, double, or triple covalent bond in a molecule, but rather a special type of dipole-dipole force centered around hydrogen atoms bonded to very electronegative atoms such as nitrogen, oxygen, and fluorine. Now recall the 3 simple molecules in the series HCl, HBr, and HI, where we confronted the unexpected result that the most polar molecule, which was HCl, had the lowest boiling point, and the least polar molecule, HI, had the highest boiling point. We concluded that for these 3 molecules, dispersion forces that scale with molar mass appeared to dominate over dipole-dipole forces that scale with dipole moment. If we add HF, hydrogen fluoride, to this series and insert the dipole moment, molar mass, and boiling point into the tables we used in slide 9, we get yet another surprise. On the left here, you can see the original table that we had for HCl, HBr, and HI, dipole moments and boiling points. And to the right, the original table we had for HCl, HBr, and HI, molar masses, boiling points. We've added HF to the top of each table. The reason we've done this, of course, is that we can now consider this a more complete series. Fluorine, chlorine, bromine, and iodine are all halogens in group 17 or 7A of the periodic table. So HF legitimately belongs in this series, and we left it out of our previous discussion, and with good reason. When we add HF to the table, it throws another wrench into our discussion. You can see that HF appears to be much more polar than HCl or the others. It also has the highest boiling point of any of the others in the series. So the boiling point of HF certainly is out of line with the others. And you can see that in the table with molar masses, HF, which has the lowest molar mass, has the highest boiling point. So what's going on here? We can look at this data graphically, and this is often done in general chemistry textbooks, by simply plotting the boiling points of this series versus the molar masses, and you see something that is not too surprising, at least as far as HCl, HBr, and HI go. As the molar masses increase, the boiling points increase, or if we go backwards, we'd say HI has the highest molar mass, HBr is a lower molar mass, so the boiling point is decreasing. HCl has the lowest molar mass, so its boiling point is the lowest. And we recall we said that it's dispersion forces that are ruling the roost in this series, HCl, HBr, and HI. So we have this decrease in boiling points, and then suddenly here's HF, totally out of line. What we say is that in this group 17 or 7A hydride series, HF, HCl, HBr, and HI, that the boiling point of HF is out of line with the others, which appears to be due to unexpectedly large dipole-dipole forces. Now, what about the other hydride series, such as, for example, group 16 or 6A, involving oxygen, sulfur, selenium, and tellurium, H2O, H2S, H2SE, H2TE, or group 15 hydride series or group 14 hydride series? How do their boiling points graph? And are there odd, out of place alignments in any of those? Well, let's look. So to the right is a plot of the boiling points versus the molar masses in this particular plot that I got from the Brown textbook. They call it molecular weight and units of AMUs, which is perfectly legitimate, same number, different units. The point is, we see the trend in boiling points versus molar masses or molecular weights for the four series. For example, the graph I showed you on the previous slide, which just involved HF, HCl, HBr, and HI, is shown on this green line right here. It's the group 7A or group 17 series. HI, HBr, not marked, HCl, not marked, and the goofy HF out of line marked right here. Interestingly, in these four series, 3 of the series show the same characteristics. In other words, 3 of the molecules line up nicely with boiling points decreasing with decreasing molar mass. But then the last one, which is actually at the top of each series, the top of the periodic table, is grossly out of line. So HF misbehaves, H2O misbehaves, NH3 misbehaves, CH4 not so much. If anything, it misbehaves in the opposite direction, but the trend seems reasonable since CH4 is the lightest molecule. So NH3, HF, and H2O are all the lightest molecules in their particular series, yet they have the highest boiling points. Note that only when hydrogen is bonded to fluorine, oxygen, or nitrogen do the boiling points show this sudden weird increase. Now, perhaps not coincidentally, fluorine, oxygen, and nitrogen are three of the most electronegative elements on the periodic table. We thus introduce hydrogen bonding, a special type of dipole-dipole force centered around hydrogen atoms bonded to very electronegative atoms such as nitrogen, oxygen, and fluorine. We can use H2O as an example of how hydrogen bonding is described. Now what we have here are two H2O molecules, the Lewis structures of two H2O molecules sitting next to each other. And what we're going to say here is that due to the very high electronegativity of oxygen, we're looking at this oxygen right here, the HO bond is very polar. In other words, oxygen's very electronegative. It pulls the shared electron pair pretty close to itself, leaving this hydrogen atom pretty exposed here. In other words, the electron it contributed to the covalent bond is pulled away from the hydrogen, leaving a fairly significant partial positive charge on this hydrogen atom. Now, this partial positive charge on the hydrogen atom is going to seek out a center of negative charge, and it finds it on another oxygen on another water molecule. So the oxygen atom in this water molecule is of course partially negative because it's pulling electron density away from these hydrogen atoms. And so this partially positive hydrogen from this very polar bond has an attraction for the negative end of this water molecule. We call this special interaction or this attraction a hydrogen bond. And the overall effect in bulk water is called hydrogen bonding. Now, hydrogen bonds are typically not nearly as strong as conventional covalent bonds. For example, estimates of the strength of this hydrogen bond between this water molecule and this water molecule is about 8 kilojoules per mole in pure water, whereas the bond energy of the actual OH covalent bond here, and of course here and here and here, is well over 400 kilojoules per mole. So in other words, the actual covalent bond is much stronger than what we are calling this hydrogen bond here. Thus, I am hesitant to call a hydrogen bond a real bond. It certainly is a bonding type interaction with another molecule, but compared to a normal covalent bond, it's quite weak. So what we're going to do is call it a special type. Hydrogen bonding we'll call a special type of dipole-dipole force. However, even though a single hydrogen bond might not have a significant strength, taken together, many hydrogen bonds can have profound effects on the properties of substances that contain them. The cumulative effects of hydrogen bonding interactions in water, for example, can be seen in the following. The first is that water has an unusually high boiling point for a molecule with such a small molar mass. The other is that when water freezes, the hydrogen bonding interactions literally force the solid state structure to be more open or less dense than liquid water. So ice turns out to be less dense than water, and ice floats on water. Now this would happen even in the absence of the usually obvious air bubbles trapped in ice. This illustration that's from the Brown textbook has the two water molecules next to each other, kind of flipped from the way I did on the previous slide. The point is we have a partially positive hydrogen atom from this very polar OH bond, which is fairly short, have a special interaction with an oxygen on a neighboring water. And this cartoon is supposed to be a cutout from this cartoon. And you can see that if you added in all the possible types of hydrogen bonding, in other words, now this hydrogen interacting with an oxygen from another water molecule through hydrogen bonding, all through these types of bond angles, it forces this open structure in ice, which turns out to be less dense than water. Now, what about hydrogen bonding involving compounds where we have NH and FH bonds? We've talked a lot about hydrogen bonding in water where we have OH bonds. Well, the intermolecular hydrogen bonding in these types of compounds can be depicted in a manner similar to water. The simplest examples would be ammonia. So we have two ammonia molecules next to each other, and HF. Here we have two HF molecules next to each other, and we see here, because of the very electronegative nitrogen in this very polar HN bond, there's a large partial positive charge on the hydrogen, which seeks out the negative end of the ammonia molecule nearby. So we have hydrogen bonding here. Same thing in HF, where the partially positive hydrogen seeks out the partially negative fluorine from a neighboring HF molecule. Now, naturally, two molecules do not have to be identical to feel the effects of significant hydrogen bonding. For example, the figure below shows two different ways that water and ammonia could interact through hydrogen bonding. So here we have a water molecule interacting with an ammonia molecule through hydrogen bonding. Notice we have hydrogen attached to oxygen, but this hydrogen being attracted to the nitrogen on the ammonia. And here's a different way these two could interact. The partially positive hydrogen on the ammonia interacting through hydrogen bonding with the partially negative end of the oxygen molecule. Now, the properties of larger molecules than these, containing OH, NH, and or FH bonds, can also be strongly affected by the presence of hydrogen bonding. Consider the isomers ethanol and dimethyl ether. Remember, they're isomers because they have identical molar masses but different structures. Now, these aren't massively large molecules, but they are larger than our initial examples. What I want to point out here is that ethanol is capable of hydrogen bonding to other ethanols. Dimethyl ether is not capable of hydrogen bonding to other dimethyl ethers because there's no OH bond here. Here there is. Here are ball and stick models and dipole moment values for each. Well, what is the effect on a macroscopic property of the capability of ethanol to hydrogen bond with itself versus dimethyl ether not being able to hydrogen bond with itself? Well, the effect is dramatic. If you compare boiling points, the boiling point of ethanol is 78 degrees C, while that of dimethyl ether is minus 24 degrees C. So a dramatic difference, which can be attributed to the fact that hydrogen bonding is available in pure ethanol, where in pure dimethyl ether, hydrogen bonding is not available. You have to be a little cautious here because you could argue that the boiling point of ethanol is higher due to its higher polarity, higher dipole-dipole forces. But the difference in dipole-dipole forces can't be that big because the difference in dipole moments is not that big. So this big of a difference in boiling points must be due to the fact that hydrogen bonding is available to ethanol, not to dimethyl ether. Now, as an aside, and not to confuse you, if we mixed ethanol and dimethyl ether, you can see that they could interact through some hydrogen bonding because you could get interaction of the hydrogen on this OH with this oxygen. So there could be some hydrogen bonding between the molecules, but that's not what we're talking about here. We're comparing pure ethanol, that means ethanol being attracted to other ethanols, versus pure dimethyl ether being attracted or not to other dimethyl ethers. Hydrogen bonding available in ethanol, pure ethanol, but not in pure dimethyl ether. Hydrogen bonding between the complementary bases, thymine and adenine, and cytosine and guanine is a major reason why DNA takes on the structure of a double helix and that it's very stable. If you wanted to draw a cartoon of the double helix of DNA, you can see there's a blue ribbon represented, which represents a sugar phosphate backbone, and there is hydrogen bonding between cytosine and guanine, C and G, or G and C, doesn't matter, T and A, or A and T, and those hydrogen bonds are what keep that double helix intact. We can show this in a little more detail if we, for example, looked at the actual interaction between thymine and adenine. You can see that in adenine there are NH bonds that can interact with oxygen on the thymine, and there are NH bonds on the thymine that can interact with the nitrogen on the adenine. Again, cytosine and guanine are capable of 3 hydrogen bonds. So you can see that the thymine-adenine interaction through hydrogen bonding and the cytosine-guanine interaction through even more hydrogen bonding is certainly capable of keeping this double helix wound. The final dipole-dipole force we talked about initially was called ion-dipole forces, which is simply the attraction between a polar molecule and an ion. And we said this type of intermolecular force is primarily responsible for the solubility of many ionic compounds, such as sodium chloride in water. We'll talk about this in much more detail in our future lectures on solutions, but for now, I'll remind you that we often write an equation for the dissolving of an ionic compound like sodium chloride in the solid form like this: NaCl solid going into water, forming aqueous sodium ions separated from aqueous chloride ions. And the cartoon here is really a good representation of what's going on when we write Na plus aqueous and Cl minus aqueous. We see for Na plus, it is surrounded, or what we say, it is solvated by polar water molecules, and we have the negative ends of the water molecules, the oxygen's end, pointing toward the positive sodium ion. We say the sodium ion is solvated by the partially negative or oxygen sides of the water molecules, and those sodium ions are kept separate in solution from the chloride ions that are solvated by the other side of the water molecules, by the positive ends or hydrogen ends of the water molecules. We will revisit ion-dipole forces and other intermolecular forces, meaning we'll bring up dipole-dipole forces and dispersion forces and so forth, the ones we covered here, when we start talking about making solutions in future lectures. Let's review. Dispersion forces are present in all molecules, both polar and nonpolar. Dispersion forces increase with increasing molar mass, or sometimes called molecular weight, and increasing polarizability of the electron clouds. And that polarizability typically increases with increasing molar mass because you have larger electron clouds, more electrons. Dipole-dipole forces are present for polar molecules, and these types of forces increase with increasing molecular polarity, measured by increasing dipole moments. Hydrogen bonding we've described as a special type of dipole-dipole force exhibited when hydrogen is bonded to a very electronegative atom like nitrogen, oxygen, or fluorine. Even though the term bonding is used, hydrogen bonds are typically not nearly as strong as typical intramolecular covalent bonds. Finally, ion-dipole forces is simply the term used for the attraction of an ion for a polar molecule.